Welcome to The Explainer. Look, today we're skipping all the cultural noise and getting straight into the microscopic, clinical reality of cannabis and the human body. We're going to take a totally objective look at how these plant molecules actually interface with our anatomy on a neurological level.
Here's the wild thing. Our brains are naturally wired with receptors designed specifically to receive the molecules found in cannabis. But modern high-potency weed is pushing this system to a dangerous breaking point. That's the central conflict we're unpacking today, right? It's literally a clash between our ancient biology and modern chemical engineering.
But before we dig into the actual mechanisms, let me challenge you with a quick question. Do you know the actual THC percentage of modern cannabis compared to the plants grown in the 1970s? Seriously, think about it for a second and keep that number in the back of your mind. Because as we explore brain biology today, the answer to that exact question explains why the modern biological impact of this plant is so remarkably severe.
Okay, let's dive into this. Here is our scientific roadmap for today. 1. The endocannabinoid system. 2. THC versus CBD pathways. 3. The developing teenage brain. And 4. Genetics and psychosis risk.
Part 1. The endocannabinoid system and our baseline biology.
All right, let's zoom way into the microscopic level to establish our baseline science. Long before humans ever stumbled upon the cannabis plant, our bodies already possessed this incredibly intricate communication network built for very similar molecules. It's called the endocannabinoid system, or the ECS. We actually only discovered it a little over 3 decades ago. It's a highly conserved lipid signaling network, and its main evolutionary gig is regulating homeostasis, basically keeping your body and brain in perfect balance. Think of it like a highly sophisticated biological thermostat that kicks on in response to stress, injury, or immune challenges. The whole system runs on cellular receptors, mostly CB1 and CB2, and the natural molecules our own bodies produce to bind to them, which we call endogenous ligands. To really get how cannabis affects us, you got to think about a classic lock-and-key mechanism. The locks are our CB1 receptors, which are heavily concentrated right up in the central nervous system. Now, your brain produces its own natural keys to fit these locks, right? The most famous one is called anandamide. It's often dubbed the bliss molecule, and it regulates everything from pain and appetite to your memory and mood. Phytocannabinoids, on the other hand, are plant-derived compounds, like THC. And here's the kicker. Because they share a remarkably similar molecular structure to our natural anandamide, they act like a biological master key. They can just sneak right in and hijack those exact same biological receptors.
Moving right along to Part 2, THC versus CBD pathways and their molecular differences.
And this brilliantly illustrates the exact mechanical differences between the two most famous cannabis compounds, THC and CBD. Get this, they share the exact same molecular formula, but they behave completely differently in the brain. If you look at what's happening at the cellular level, THC acts as a direct, partial agonist. It locks perfectly onto the primary orthosteric side of the CB1 receptor. And when it does that, bam! It triggers a whole cascade, specifically inhibiting adenylyl cyclase and dropping cyclic AMP levels. That drastically alters neuronal excitability and neurotransmitter release. And unlike our natural anandamide, which does its job and breaks down quickly, THC has a much longer residence time. It just hangs out, forcing the signal to stay on and completely overwhelming the system.
So you can really see the disruptive nature of THC when you contrast it directly with CBD. While THC binds right to that main receptor site, causing intoxication, distorting time, and significantly impairing short-term memory encoding in the hippocampus, CBD takes a totally indirect route. CBD is what we call a negative allosteric modulator. It binds to a completely separate site on the receptor and actually changes the physical shape of the receptor itself. It's kind of like a dimmer switch. It doesn't activate the receptor on its own, instead it modulates the whole pathway and actually turns down the potency and efficacy of agonists like THC.
All right, Part 3. The developing teenage brain and their real-world neurological impacts.
Let's zoom out from those microscopic cells for a second and look at how these molecular disruptions impact human neurological development, especially in kids. According to recent clinical data, adolescent cannabis abuse in the U.S. has spiked by a staggering 245% since the year 2000. That is huge. And hey, remember that question I asked you earlier about modern THC potency? Well, the rise of high-potency products like dabs, wax pens, and concentrates means today's teens are being exposed to THC levels that absolutely dwarf anything we saw in previous decades. They are heavily, heavily flooding their developing endocannabinoid systems.
So the crucial point is this. The human brain doesn't actually finish developing until you're in your mid-20s. Flooding a maturing brain with high-potency THC has some very real, severe clinical impacts. Research shows it can literally reduce the production of dopamine in the mesolimbic pathway, which completely blunts motivation and reward processing. It disrupts the hippocampus, totally impairing learning and memory. It alters the prefrontal cortex, the area responsible for decision-making and impulse control. Ultimately, all of this intense overstimulation of a developing endocannabinoid system leaves the teenage brain highly vulnerable to addiction as well as anxiety and panic disorders.
Which brings us to part 4, genetics and psychosis risk.
Now, this brings up an absolutely critical question. Why do some users experience severe mental health outcomes while others are totally fine? To answer that, we have to widen the angle and look at population-level genetics. The answer lies in what clinicians call a gene-environment interaction. Basically, your DNA creates a biological predisposition, and the environmental factor, in this case the high-potency THC, acts as the trigger. Researchers have actually pinpointed specific genetic variations, like the COMT valine 158 allele and mutations in the CNR1 gene, which is the gene that actually codes for the CB1 receptor itself. Folks carrying these specific genetic markers are significantly more susceptible to cannabis-induced psychosis, or CIP, when they're exposed to THC.
Now, what's really interesting about this slide is the stark clinical data on the distribution of that CNR1 gene. Look at the group with cannabis-induced psychosis, that's group C0. A massive 72.7% of those patients possess the homogeneous allele of the CNR1 mutation. Let's compare that to patients with schizophrenia who had never even used cannabis. In that group, this specific genetic expression was found in only 3.3%. Wow, that dramatically underscores how having a specific genetic vulnerability, when you combine it with cannabis use, radically amplifies the risk of psychotic disorders.
So when we connect the microreceptors with the macrogenetics, we start to see a really clear picture of the modern danger here. Our endocannabinoid system is perfectly designed to maintain equilibrium using just small, short-lived bursts of natural endocannabinoids. But modern, high-potency THC products act like a massive stress test on this system, completely overwhelming our natural homeostatic limits. For people with those underlying genetic vulnerabilities we just talked about, this systemic overload can literally break the brain's regulatory mechanisms, leading to severe outcomes like psychosis and schizophrenia.
Which really leaves us with a provocative thought about the future. As high-potency cannabis becomes totally mainstream, will genetic screening become a necessary prerequisite just for informed use? For unfiltered, evidence-based science on the things that shape our world, click that subscribe button now. Thanks so much for hanging out and exploring the incredibly complex biology behind the endocannabinoid system with me. Keep asking questions, keep looking closely at the data, and I'll see you in the next explainer.